everyone. I welcome you to the second day of our conference. I hope you had a wonderful time in the early session. And after the presentation by Wolf Geppert, we will have the later session of this day. Right now, we are waiting for our keynote speaker, so there will be a delay. But to not lose time, I will tell you a little bit about how the conference platform works and what kind of options you can use there. So it will be even more entertaining during the rest of the day and also tomorrow. So today morning we had three interesting sessions: the extremophiles, the education and space education, and also we had the origin of life session. So if you missed any of the talks, and I know it was impossible to be on two talks at the same time, we are recording the talks, and the system itself, the platform Hopin, is rendering it and will be putting it under replay button on the left side of your platform. It will be done with the delay, so we expecting the talks from yesterday to appear sometime today. I checked them about 5-10 minutes ago. They weren't there yet, but after the conference is finished, they will definitely be there, and you can still log in to the conference platform after the event is done, and you will still be able to go back to and revisit those talks that you wasn't able to see. Another interesting feature that we use for the networking breaks, and some people ask me how to find it, is on the left side menu. There is the networking, and then if you press the button like to join the networking session, it will automatically assign another person waiting to be connected, and you will have a one-on-one session that lasts, I think, 4 minutes. And during that time, you have some private time to chat about what happened at the conference. The more of them you do, the more people you will be able to meet. So I highly encourage you to do that. We also have the chats. There is this main chat for the whole conference, and be careful when you're writing in a chat. There is several chats. So if you're in a session, you're on a session chat, but you still have access from the menu to the whole event chat. So where you can send a message to everyone in the event or everyone in the session. So that is also very helpful. Right now we are on the stage. That's probably where you're watching me. And as I said, we have a slight delay waiting for our key speaker to appear, and it might take another 10 to 20 minutes. So please stay with us, and we'll start as soon as we are ready. And for now, if you would like to ask any questions, this is a good time to ask them on the general chat, or just say some words of appreciation to anyone that presented today, or ask some additional questions. If we communicate now on the event chat, it will be visible for everyone. Yes, as I said, our stream will start as soon as we have our key speaker present. We are reaching out several ways, so just a little bit of patience. And well, this is so typical for online conferences, but doesn't really matter because we are still recording that, and we will be able to start whenever our key speaker is present. For your information, you can also see the stream of any keynote speakers. And also later today, we have the panel discussion about the artificial Intelligence and a Search for Life that will be transmitted just as this talk is going to be transmitted. And as you are right now listening to me, it will be also broadcasted, and it is live broadcasted to Facebook and YouTube, and also on our Hopin platform where majority of you is probably watching. But there is, and it is an open event, so anyone in the internet can see it. So it's on our YouTube channel uh, of the Polish Astrobiology Society. I will try to find a link and paste it on the chat. So if you want to share with your friends, you will be able to use that to send on your social media. Maybe you already seen the Facebook event. So we have a Facebook event for this conference, and you can easily share that event with your friends. And as I mentioned, the key speakers and the discussion later today are available for anyone outside of the conference. For anyone that is late, we are still waiting for our key speaker, and it's probably gonna take a little bit more time. And I'm right now searching for a link that I can share with you.
Probably
For
gonna take a little bit
I
more
already found
time.
it. For YouTube that you can share with your friends, especially for the keynote sessions like this one. Also, tomorrow, Lynn Rothschild is going to present about this use of synthetic biology in space exploration. So once again, I just shared a YouTube link on the chat of Hopin platform while we're waiting for our key speaker that unfortunately is still not present at the platform. So it might take more time, but stay with us. Maybe grab a cup of tea or coffee, depending on the time of the day, and yeah, use this time as you want. We will be online when the key speaker shows up. I mean, I'm online here anyway. Oh, not sure if that is gonna help. Can you copy it from the platform? Oh, maybe if we could copy a QR code, that would be useful. For the event on Facebook, I'm gonna show that too. Yes, so we shared that on Hopin, but we also need to share the Facebook link for the event on Hopin platform in the main chat. So if someone could help me with that, I will be looking for that in the meantime anyway. For anyone who's going to present tomorrow or later today, it is very useful to get to first enter the session where you can practice. So in the sessions, there is a room called preparation room for speakers, and there you can check if the sharing the slides works correctly. I know it is quite internet demanding. So some people had issues today with the connection, but eventually everything went well. So be patient. This kind of a platform of an online conference is usually more challenging than in person, but there are some amazing advantages. We did not have to travel to anywhere to be able to see it. And also, uh, yes, well, with the connections all around the world with a very low cost, I think it's something that pandemics taught us. I hear Priya suggested that you would like to hear about the research that I do. Uh, I think Part of it is going to be covered by Lynn tomorrow. So that will be an interesting part of a synthetic biology. There will be a summary. Mm, but also, if we're talking about my current projects at NASA Ames, I am working on a very interesting protein aggregation in the form of amyloids. Uh, that is a very concentrated aggregate of proteins in a linear form. It appears in, you might know these from the Alzheimer, Parkinson, Huntington, and other diseases, that the amyloids are part of aggregates found in human brains. But we started studying this kind of a protein behavior in ancient organisms, like in a well-conserved phylogenetic lines like are in archaea and cyanobacteria, and we found some candidates, and we also confirmed some of archaeal protein to be able to form those fibers that can not only be used outside of the cell to aggregate them together, but they can also work as a memory molecules, providing the means for epigenetic inheritance on the form of aggregated protein. So after doing that in archaea, we confirmed that this kind of a protein behavior is present not only in uh, in bacteria and eukaryotes, but also in archaea. So all across three domains of life, and that makes possibility that this kind of aggregates are even older than the branching of these three domains of life. So maybe reaching Luca or even like the beginning of the origin of life. And so our methods that we were using are the top-down approach. So we searched in the genomes of contemporary organisms, but the next step is to use a bottom-up approach where we use an unbiased library of peptides, and we will see how many of them are able to form this kind of aggregates spontaneously. We know that short peptides can be synthesized abiotically on clay, for example. So they could have been forming even before the first life started, and maybe the life had to live in the context of aggregating proteins in the form of amyloids since the very beginning. And at first, the anti aggregation systems in the cells were not efficient, and it was a detrimental threat when a protein aggregated in this way. But then later, with development and evolution of anti-aggregation system in eukaryotes, they actually learned, especially in yeast, we see that how to use those aggregates for their benefits and as a second mode of inheritance. So that's a short summary of a pre-and research that I'm doing. I'm also working right now for a startup that tries to substitute chemical fertilizers with microorganisms that are modified to interact with the root of the plant and to release ammonia. So a very sustainable ecological way to fertilize, because as you probably know, and we heard even in on this conference that production of biofertilizer is a biosignature, is a techno, could be a techno signature. That was a presentation from yesterday from Jacob. He did mention that how population on human population on Earth is growing in correlation with the amount of fertilizer, chemical fertilizer used, but unfortunately, it's very unsustainable form of food production. It depletes the biodiversity. It is a source of one of the gases that is a major contributor to ozone layer depletion and also an incredibly efficient housewarming gas. So this kind of microbes that we're trying to build are going to be useful not only here on Earth, but also if you imagine space travel, uh, with the fertilizer, it is so costly to bring anything on the orbit or even farther. So taking a little bit of microorganisms is a premise of the whole synthetic biology field. You take a microorganism that you can then grow on a space station or on a moon base or Mars base, and then you just using the in situ resource utilization approach, build more of your microbe. And today we will have another session that is called the extraterrestrial habitation. We will have three talks. One of them is not going to appear, so we have only three talks, but very fascinating subjects of how to use um, biology production, but also we'll have a talk about architecture, and these are the main subjects that will be covered on the extraterrestrial habitation session. That will be very interesting to see. But the parallel sessions about the astrochemistry and then artificial intelligence, they all tie into use of AI in the research. And right after that session, we will have the AI panel. Our keynote speaker is still not present, so we are still patiently waiting, and I am so grateful that you're still with us after almost 17 minutes of delay. But let's wait and see what happens. I will take a little break and see on my other channels to see what is the progress on the communication with our keynote speaker for today. So give me a few seconds. So unfortunately, we still cannot reach our keynote speaker. And just to use this time and this opportunity, as I am still able to talk to you and you're still listening, thank you so much for being here and waiting patiently with me. I might actually look for some slides and tell you more about the research we've been doing at NASA Ames about prions, and maybe introduce you a little bit more to this subject, because just a few minutes before I was telling you, oh, 1 second, because I'm finding those presentations on the spot is not that easy. So yes, you already learned about amyloids, then the prions are a subtype of those amyloids, and it would be very useful if I could show a presentation at the same time. So bear with me, I will bring it up. Okay, I have the folder with presentations, and it will take a moment. I could also tell you more about just the theories in the origin of life as a general subject. But let's go for prions. Okay. I will share, first this is done, and I just need to share my screen. Mikołaj, you are going to be on the backstage, so I will, when I'm presenting, I'm not gonna see if Wolf showed up, our key speaker for today. I will be substituting him right now with the presentation, but please interrupt me anytime when the Wolf shows up, and I will finish it another time. All right, so now you should see the screen, and I will tell you about the evolution of the perspective on prion-based mechanism of regulation and adaptation, and what is connecting cannibals and the space exploration. That is a challenging leap to do, but where you're looking at right now is a alien island of Papua New Guinea, where Daniel Gajdusek discovered a tribe of cannibals that suffered from a weird neurological disease that seemed to be transmitted from a person to another. And at that time, no neurological disease of that sort was known. And on this image, you can see kids and you can see women because they were the one that were affected by this disease. Mostly, they were the one that were eating brains, and that's why the sticks that they hold is because they experience tremors and problems with standing still. Um, and when we're looking at prion, it's probably best to start with the mad cow disease because the term prion became recognizable by the general public because of this disease. But during this talk, I'd like to show you that not all prions are bad and they're very common in nature. So prions attracted the global attention at the end of the last century when the epidemic of prion disease broke out in Great Britain. This epidemic was very unusual because it could spread between species and precisely from cows to humans. And so far, there were two Nobel Prize awarded for the study on prions. One for discovery of the novel way of disseminating diseases. This is what Daniel Gajdusek discovered on Papua New Guinea. And the second one for identifying the actual infectious agent as a something that was even smaller than a virus. So the experiment showed that if you deplete the material, infectious material of nucleic acids, it still causes the disease. And it baffled scientists for a long time. So what was the infectious particle was a protein, and this protein was eventually named prion protein. So it is, and the name prion protein, prion comes from proteinaceous infectious particle. It's a kind of an acronym. So the scientific community was reluctant to accept the discovery of an infectious agent made only of a protein. None of that was known before. And prions are still the only known infectious agents that have no nucleic acids. This difference makes prion replication fundamentally different from all biological processes. And the fact that the protein can store biological information seems to even contradict one of the major our understanding of the information transfer in a biological system, so-called central dogma of molecular biology. And this uniqueness of the prion can also promise some amazing discoveries in the fundamentals of how the biology works if we don't need the nucleic acid to transfer biological information. But only now we just begin to understand how important the phenomenon is in a normal biology, not in the disease, but in an everyday life of a cell. So this animation shows a formation of the original prion that causes mad cow disease. So a prion protein forms a prion. I know it's a little bit of a tongue breaker, but that's what is happening here. So at the beginning, the protein changes its secondary structure, increasing its content of beta sheets. The misfolded protein is then interacting with other copies of the same protein, changing it into the same configuration. And this process repeats indefinitely until all the soluble forms of a protein is depleted. So they're building a higher order structure that grows in one dimension in the form of fiber, and such fiber are shown on the right side of the slide from the side. Right? So it's just like a ladder. So most known prions, they can acquire the form of amyloid. So the amyloid that I mentioned before for Alzheimer, Parkinson, and Huntington disease, and many others, those and are a structural form that the prion can take, but not all amyloids are going to be prions. Only a subtype of them will behave in this specific way that it allows them to be inherited between cells or between organisms. So importantly, the amyloids that are built out of one and the same protein can take slightly different shapes. And you can see this A, B, C, D forms. These are fibers that are made of the same protein, but they do look different. And the shape depends on the final conformation of the protein that is building it. And interestingly, different shapes of the fiber have different impact on the infectivity or the ability to cross species barrier. So the conclusion from this is that the biological information is stored in the amyloid fiber shape itself. And here is a schematics that shows the lifetime of a aggregate. So you start with the monomers, then you build an oligomer. You still have a chance to go back to the monomer, but at some point when the protofiber is formed, this integration of new parts become extremely fast. And in a logarithmic phase, you end up with amyloid that is steady and stable, matured. And because there is no more monomers available, this line is flattens down. So prion relationship with amyloid doesn't really help to repair the image of prions as being bad, because they're connected to Alzheimer, to spongiform encephalopathies, prion disease, dementia, Parkinsonism, amyotrophic lateral sclerosis, Huntington disease, spinocerebellar ataxia, and also even type 2 diabetes or systemic amyloids, and many more. So these are all nasty diseases, and that's how we discovered amyloids. But are they also present in other organisms and more physiological terms? Yes, they are. So, for example, amyloid form is used. Different proteins can build amyloids. So, peptide hormones, for example, are stored as amyloids. Melanin in our skin is scaffolded as an amyloid. Well, amyloid is also an important part of a structure of silks, for example, in silk or in a spider web. Amyloids are also used by bacteria. Proteins excreted by bacteria very often form amyloids, and the most known of them is a Kerley protein, or CSGA, a Kerley fiber produced by very commonly used Escherichia coli, a model organism in many labs. But also Staphylococcus and Bacillus, they have proteins that are excreted and form amyloids. There are some proteins in humans that if you remove the ability of forming amyloids, they would compromise immune response, because they actually boost up immune response by aggregating. There are also two examples of proteins that are evolutionary conserved between Drosophila, mouse, and human, and they all responsible for long-term memory formation. If you're not able to aggregate in the form of amyloid, this protein, if it's not able, then the long-term memory is not being formed in mouse. So very exciting discovery. Biofilm is another part in which amyloids are very useful. So biofilm is a conglomerate of cells bound in extracellular matrix. In the form of biofilm, bacteria are more resistant to stressors, and they can attach strongly to different surfaces. This extracellular matrix that is composed of many different substrates, but including amyloids, helps them survive and stick to different surfaces. And biofilms, they can cause problems. So for example, in medicine, they are present in the mucosal surfaces, adhere. That's where a lot of infections start. In the technical systems, they cause so-called biofouling or biodegradation or biocorrosion. In space, there are documented examples that the biofilm caused troubles on International Space Station and other spacecrafts. So it mostly leads to the degradation of components of the systems or cause short circuits. Here you have some photos from an actual ISS port with this nasty biofilm forming in a tubes. And the more we fly into space, the more we will bring our bacteria, and these bacteria are gonna, the older the systems are, and we give more time for bacteria to grow, the more of a problem it is going to be. So there is a big emphasis at NASA to work on mitigation of the biofouling on ISS, for example, and we suggested a proposal to how to use anti-amyloid reagents to stop forming amyloids in the biofilm. So, for example, here you have a growing colony producing more and more of an extracellular matrix that includes amyloids, and the web of those interactions keep bacteria together. But if we are able to use, for example, surfaces that would be covered with anti-amyloid compounds, those bacteria should not that easily stick to the surfaces. And on the right side, there is a list of potential reagents that we could use to do that. Now I would like to bring you to even more maybe complicated, but like there's a conceptual shift that we have to do because I'm using the term amyloid and prion, and there is a slight difference that might be hard to grasp, but this graph should help with that. So first of all, the functional amyloid, so what the bacteria are using, for example, to create a biofilm. These are proteins that are synthesized inside of the cell, and they don't have really a function inside of the cell until they get exported and they convert it into amyloid. Then we have this functional amyloid that helps, for example, sticking cells together in the form of biofilm. The prion is also making an amyloid, but the situation is dramatically different. The protein synthesized is a functional protein that has some specific, they're very different, there could be very different functions depending on what kind of a protein we're talking about, and we identified thousands of proteins in bacterial genomes that can form amyloids inside of the cell. So at first, those proteins that are usually, for example, like gene regulators or like translation regulators, they function normally, but then they can experience this conformational change that would make them stick together and form an amyloid fiber that deactivates their first function. Moreover, this formed amyloid can be passed to another cell, and during division or when the cell breaks up, these fibers would stay in the environment. They're extremely stable. That is also important to know. This is why they're used in the biofilm, because the aggregate in the form of amyloid is extremely resistant to different pHs, temperatures, and just physical sharing. So a major difference between amyloid and prion is that amyloid is functional amyloid is gaining function when it aggregates. A prion is usually losing, the protein that builds a prion is losing its function. And we can predict new prions. So proteins, as we look at the known prion proteins, and the majority of them are right now known in yeast, we can see that there is some common characteristics. So they're usually a low complexity, that those proteins include low complexity regions. Those regions of protein that is responsible for aggregation in the form of amyloid/prion are glutamine and asparagine rich. There is a depletion of charge and hydrophobic residues in this protein fragment. and and there are long intrinsically disordered regions that are also important for a phase transition. There is a lot of research right now on phase transition of disordered proteins. So the subject of prions strongly overlaps with the phase transition too. So here are some examples of proteins in different organisms that were confirmed, verified experimentally as prions. So in Saccharomyces cerevisiae, so the common yeast, uh, there is more than 30 prions already known. Probably Saccharomyces cerevisiae is in general the best studied organism on Earth. So it comes not as a surprise that also the major number of confirmed prions it was found there, but we're slowly expanding to other organisms. An interesting example that I didn't mention yet is in Arabidopsis, where a lumen-dependent protein is forming a prion, and its ability to form a prion is responsible for flowering, so a very physiological process. And here's how the life cycle of a prion could look like. So as we mentioned, like on the slide with the graph, we start with the native single monomer conformation of the protein that spontaneously misfolds and then converses other kinds, like other copies of this native protein into a similar beta sheet rich protein structure. And then this process called nucleation leads to the formation of a prion seed. And from that point, everything goes really fast. The long amyloid fiber is formed. And then if the fiber breaks, one of the most popular theories on how they're formed is right now focusing on uh, aggregating at the ends of the growing fiber. So when the fiber breaks, you get twice as many ends to interact, and then it breaks again and again, and that's why it becomes exponentially fast. Also, this breaking can be facilitated by chaperones. So chaperones are extremely important for prion dissemination. So actually, if you inhibit chaperones, you are going to see that the prion uh, is no longer being transmitted to another cell because the fiber becomes too stable, it's not fragmented, so it doesn't find a way to another cell. And that's one of the way how to confirm that the protein is behaving as a prion, because you can stop the transmission by influencing chaperones. And of course, I had to put the former governor of California on this slide. So because prions can be lost, but they can also be cured. And as we say, they will, after being cured, they will be back. So these are three things that characterize prions: a reversible curing. So first you have a normal form of a protein, then spontaneous transformation into prion form that happens once every million division of cell. They were talking about yeast in this case. Then you can use curing by influencing chaperones, for example, and they will go back to the natural native form of the protein. But after 1 million divisions, they're going to again create a prion. So prion is a persisting behavior of a protein. If it can form a prion, it will, and it depends on how many of the proteins is being produced. And this is the second example. If you have a standard 1 million division per prion, if you increase the number and expression of the protein, and therefore the number of copies of a protein that can experience this behavior of forming a prion, you will get prions more often. Also, another way to distinguish a prion is that the normal protein and the no protein phenotypes are very different from each other. And when you have a prion phenotype, it looks like a deletion of the gene. So what I was showing in a few slides ago is that the prion and the protein that forms a prion has a function, but when it aggregates, it loses its function. And the loss of function could be resemblant to the mutation where you deleted the gene that codes for this protein. So you still have the gene that makes the protein in the case of prion, but it's aggregated non-functional, so the phenotypically it looks like you deleted the gene. Here is some nomenclature about the prion, and that's how we designate with the big letters because it's a dominant phenotype. We add plus because it's a cytoplasmic element, and we have it in the brackets because they don't obey Mendel laws. And a comparison non-propagating form of a protein would be named like that in a small letter case and with a minus. So prions, in summary, behave like a heritable molecular switch, and the rate of the on and off is evolutionary balanced or tuned. And so prions could be called the transmissible aggregates that change phenotypes. And this is a very striking example where you have a non-prion form of a red pigment forming, creating yeast. And then when you acquire a prion form, it turns into white. And if you look closely on the left side, some of the cells do not have the pigment because 1 in a million, 1 in 100,000 generations, in this case, the cells are going to acquire the prion form that is phenotypically white, and then you can just transfer it to another plate, and you will grow the whole population of white cells that eventually are going to lose the prion and turn red again. Under the microscope, you can see that a non-prion form has proteins that are soluble in the cytoplasm, and when they aggregate, they form foci that are totally visible under fluorescent microscope in yeast. So these are pretty big, stable aggregates. All right, but what is going on there under the hood of the cell? So why do we have those red and white colonies? So on the left side, you see the red pigmented yeast, and on the right side is the white pigmented yeast. The one on the left has a protein in its normal soluble form, and on the right side, it is in the aggregated amyloid forming form of the protein. The protein that we're talking about right now is a protein SUP35. And the SUP35 protein is a repressor of translation. It takes part in the formation of a repressor. It's part of a complex. So these little dots are the SUP35. This is another protein that it binds to, and then together they recognize the stop codons and they inhibit translation. So the ribosome going on the mRNA, it's going to stop at this stop codon because it was bound to the repressor. And that's the normal situation in this case. On the other hand, here, the protein SUP35 is forming an amyloid, so it's not available to form the functional repressor. So the ribosome is not so efficiently recognizing stop codon, and it's actually going to make a longer protein. This is a little bit convoluted example also because we are looking at the mutant strain of yeast that is very practical for prion research. By on default, this gene has a premature stop codon, and this premature stop codon doesn't allow the cells to grow without adenine. It's a gene that helps to produce adenine. And the byproduct of not being able to produce adenine is red pigment that is accumulated in a cell. So these cells on the left, very conveniently, are not going to be able to grow on adenine, but those on the right are going to be able to grow on adenine missing media. So that's a media that is depleted in adenine, but it's still, the cell can produce the whole genes, the proteins that are needed in adenine synthesis. So two things: color change and also ability to grow an adenine. So what is going on here after the stop codon? we're using this example for adenine, but it could be any other gene. So if you have a situation in which and normally the stop codon is recognized, what is going on this part of the gene? There is no evolutionary pressure, and it could be anything that is being produced after that. So this actually allows evolution to speed up in these genes after the stop codon, and when the prion appears or disappears, makes it available or not for reading, it could produce any kind of a result and an exploration of new forms of proteins. And the prions that we studied and that we scanned the whole genomes of many proteins, like the full proteomes of bacteria, archaea, and eukaryotes. And what we found that majority of prion-forming proteins are translation terminators, are transcription repressors of multigenes. They're chromatin remodeling proteins. They're RNA binding proteins. So it looks like they're working an epigenetic, over or outside around the gene inheritance. So they seem to be working on top of a traditional genetics, because any of these, if they aggregate, they're going to influence function of so many different genes. If you, for example, mess up with the translation terminator, as in a previous slide, it's not going to affect just one protein. It's going to affect all of the proteins. So the phenotypic change could be vast. And this is how it could be useful in evolution. You start with a cell that doesn't have a prion and lives in the environment number 1, that's the green field. The environment number 2 is going to be the blue field. So we go from a non-prion form of yeast, and at some point they do acquire, some part of them are acquiring this prion phenotype. But then the environment changes, and let's imagine the situation in which the natural state is not permissive for this environment. The cells would normally die, but only the strongly changed phenotype of a prion-bearing cell is able to survive and rebuild the population. And as we said, they can reverse back to the non-prion state, but they also had time to acquire a classical genetic mutations that could actually help them survive in the environment number 2 and rebuild the population. The prion is lost, they rebuild the population with the standard form of a cell, but now it is already evolved to be able to survive in environment number 2. So prions could be evolutionary capacitors, and we call this process biological bed hedging. It is a term from gambling, where you put your little dab on something that is very unlikely to win, but if you win, you can score a lot, and that's what cells are doing. So the prions seem to be evolutionary capacitors. The non-prion state, the untranslated region can accumulate genetic variations. In a prion state, polymorphs become phenotypically expressed, and in the prions, if the prion state lasts long, the population expands and mutates, and those mutations allow cells to maintain traits even after the prion is lost. So it seems like a little bit of inheritance of experience. So a very Lamarckian view. So the prion mechanisms, they can integrate diverse inputs, and then they can actuate diverse outputs. They provide immediate access to genetically complex traits. And so as Daniel Jarosz, my supervisor at Stanford, said, the controlling of the protein organization using prion-like behavior is a new paradigm in biochemical regulation at the single cell level. So yes, it also gives us a new genetic or epigenetic tool to control expression of many genes at the same time. So I hope that in this presentation I was able to show you that there have been a shift in the perspective on prions from rare and infectious agents to very common with multiple important functions. So, how old are the prions? This is the question that I started asking at NASA. Could it be that they were present on Earth very early in the life's evolution? And so the question of how old are prions boils down to the question how old are the amyloids, because the amyloids is this physical form of a protein, of a prion protein anyway. So if we look at that, we found amyloids in all domains of life. So pretty ancient, probably it is. It also, it's mostly made of beta sheet, and beta sheet structure of protein is considered a very ancient structure of the protein. If we look on the ribosome, the beta sheets are in the first proteins on its outside shell. Then amyloid also is seen as the lowest energy state possible. That's what this graph shows, the energy landscape for protein folding, where you would have an amorphous aggregate here, you would have a well-folded protein on the left side, and as you can see, the amyloid fiber is the lowest energy that the protein can even get. And importantly, the amyloid can be formed out of very short peptides. Like in this case, this is an electron microscopy of just this peptide that is maybe 9 amino acids long. But the shortest ever done was just 2 amino acid long. So very basic structure as it seems. So in our research, we're using the bottom-up approach in which we are mixing RNA coding for random from random peptides, a whole library of it, and we mix it with the PureFrax, that is a cell-free expression system, and we encapsulate it in liposomes to form amyloids and then to recognize them and filter them out and go back to the sequence of the RNA and learn what kind of very simple peptides could actually form amyloids. So on the other hand, the top-down approach is to look at the contemporary organisms and their DNA and search in their proteomes for proteins that resemble amyloid aggregates. And for that, we're using artificial intelligence algorithms that resemble artificial intelligence. So at the beginning of my project, when I was looking through the literature, I found out that prions have so far been verified in eukaryotes, and only 2 of them were found in bacteria, but none was found in archaea. And with this little number of examples, it was impossible to draw conclusions about the phylogenetic relationship of different prions. So our priority should be to find more prions to be able to assess their evolutionary origins. And my ideas caught the attention of evolutionary biologist Lynn Rothschild, who became my supervisor at NASA. And this is how we started our hunt for prions in archaea. So at first, we built this phylogenetic map of candidates for prions in archaea. Some of the species had more than 15 prion candidates per genome. Some of them had less, but in general, it was almost 3 million proteins. And out of analyzed, and out of them, about 0.1% were prion candidates. Then we went to the lab and an experimental verification and a test. We tested 16 proteins, and 8 of them formed amyloids, and 6 of them were able to carry information by recreating non-Mendelian patterns of inheritance in yeast when we expressed them in yeast. So in the first experiment, I used a method in which the candidate protein was expressed and exported outside of E. coli. The E. coli was growing on a media supplemented with Congo red, and the Congo red is a dye that specifically binds amyloids, causing them to turn red. The colonies turn red. So it was a simple system. And then the first level of our experimental screen to see if a protein is capable of forming an amyloid maybe just acquires color for now. But then, using the electron microscopy, I examined the morphology of the aggregates formed by prion domains, so the fragments of a protein that are responsible for the aggregation. And I noticed that 8 out of 16 tested indeed formed amyloid fibers. And consequently, using several spectroscopic methods, we confirmed that aggregation kinetics is typical for amyloid, this short lag phase, exponential phase, and the plateau. And we also confirmed a high content of beta sheet that are very typical for amyloids. And for that, we collaborated with the University of Warsaw and Robert Detz, that is also on this conference. In this assay, 6 candidates were able to create prions. And so this is a yeast assay where I expressed heterologically the prion candidates in yeast in this mutant strain that acquires colors. And we were able to create a prion which was trackable because it allowed cells to grow on adenine-free media, and the colonies obtained a very characteristic red coloration. In conclusion, our experiments demonstrated for the first time that the proteomes of archaea, you can find proteins that can form prions in archaea. And we pretty much just filled out this dot. Now we know that our observation complemented the picture of prion distribution within the main branches of evolutionary tree of life, and suggests that there might be a continuity between the first abiotic amyloids and the modern prions that act as regulators of numerous biochemical processes. And unfortunately, sometimes they contribute to pathological conditions such as prion disease in humans. And right now, we're focusing on the oldest prions on Earth. We already know they exist in all 3 domains of life, and the paper that I'm trying to submit before Christmas is presenting a list of potential proteins that are potential prion proteins that are conserved across all 3 domains of life. It wasn't many, but we were able to find proteins that had identical functions across all 3 domains of life, and they all have prion domains. So very interesting candidates. So prions and the spectrum of functions they comprise likely represent one of the largest paradigm shifts concerning molecular encoded phenotypic diversity since identification of DNA as a principal molecule of heredity. This is a quote. It is very, it's a bold statement, but unfortunately it's not me. I can just quote someone. But yes, I think about it the same. I think it's one of the biggest shift in our understanding of inheritance of phenotypes since we discovered the DNA. And also, of course, I am a president of a Polish Astrobiology Society, to which we are grateful for organizing this conference. And please visit our website, AstroBioPL. And with that, I would like to thank all my collaborators and friends. This is a photograph from Ames Research Center in 2018 when I first joined the team. So thank you so much, and this is the end of my presentation. I quit the panel so I can see that unfortunately our keynote speaker is not yet present, and I think we are slowly getting to the end of the time of this session, but I hope at least I could substitute partially the absence of the key speaker for today. Yes. All right, let me check on the time for the sessions today.